Assalamualaikum and hi. So today I will start this presentation with introductions. I will be presenting a detailed case study on animal reproductions, focusing on an unusual reproductive phenomenon observed in Komodo dragons. Komodo dragons, or scientific name is known as Varanus komodoensis, are the largest living lizards and are native to Indonesia. They are typically known to reproduce through sexual reproduction, which is involving male and female individuals. However, some researchers discovered that some female Komodo dragons were able to produce offspring even when they had no access to males. This discovery was made in controlled environments such as zoological parks and also conservation centers. Genetic testing confirmed that these offspring inherited genetic material only from the mother. So, this reproductive phenomenon is known as parthenogenesis. The discovery is scientifically important because it challenges traditional biological assumptions about vertebrate reproductions, including reproductive strategies, meiosis, genetics, adaptation, and evolution. Okay, so next I will go with the definition of parthenogenesis. What is the parthenogenesis? Parthenogenesis is a form of asexual reproduction in which an embryo develops from an unfertilized egg, meaning that no male gamete is involved in the reproductive process. In this type of reproduction, the offspring inherits genetic material only from the mother, which is from the female. This process is different from sexual reproductions, which requires the fusion of male and female gametes and results in genetic variation among offspring. Parthenogenesis is commonly found in invertebrates such as insects, but is considered rare in vertebrates. However, studies such as the ResearchGates report that Komodo dragons have shown that parthenogenesis can also occur in large vertebrates, especially when females are isolated from males. This demonstrates that parthenogenesis is an alternative reproductive strategy rather than a completely separate reproductive system.
There are 6 types of mechanisms or parthenogenesis. First, automesis. Automesis is post-meiotic process in which a haploid cell may either duplicate its chromosomes or join with another haploid cell. This mechanism can lead to offspring that are genetically similar to the mother but may also involve some genetic recombination. One example animal that have this type of mechanism is Komodo dragons. Second, apomesis. Apomesis bypasses meiosis together. This mechanism allowing the development of the egg directly from a diploid cell through mitosis. Apomesis occurs in some rotifers as well as in insects and flowering plants. Third, gynogenesis. Gynogenesis is a form of parthenogenesis where sperm is involved only for activation of the egg. This mechanism occurs in some species of fish such as Gambusia species. Fourth, aerhinotachy. Aerhinotachy is the development of male offspring from unfertilized eggs. This mechanism is seen in several species of insects such as honeybees, where drones are produced from unfertilized eggs laid by the queen. The presence or absence of the fertilizations determines the sex of offspring. Fifth, teletochy. Teletochy is the development of female offspring from unfertilized eggs. In species exhibiting teletochy, female reproduces sexually and all offspring produced by this method are females. This mechanism is seen in certain reptiles, amphibians, and invertebrates and some other species such as aphids. Last, deuterotachy. Deuterotachy is the development of both male and female offspring from unfertilized eggs. This mechanism depends on the environmental stress. Deuterotachy is observed in wasps and aphids. Let's talk about parthenogenesis in Komodo dragons. Komodo dragon is the largest parthenogenetic lizard in the world. The Komodo dragon's parthenogenesis secret was uncovered in 2006 at Chester Zoo in England. This secret is revealed when female Komodo dragon Flora has laid a clutch of viable eggs without any interaction with the male Komodo dragon. After the incident, The study on reproduction system of female Komodo dragons was conducted. According to Ahyari Hananto, female Komodo dragons produces offspring without male fertilization. The genetic mechanism of parthenogenesis in Komodo dragon is automixis. In Komodo females, each egg contains either a W or a Z. Hence, parthenogenesis leads to embryos that are either WW or ZZ. Eggs that consist of WW material are not viable and die off. In contrast, ZZ does work, so all the parthenogenesis Komodo's babies are male.
This is the genetic consequences of the parthenogenesis. Number 1. Reduce genetic diversity. Parthenogenesis produces offspring from a single female without genetic contribution from a male, resulting in little to no genetic recombination. Hence, offspring are genetically identical or very similar to their mother, leading to reduced genetic diversity within the population. Reduced genetic diversity limits a population's ability to adapt with the environmental changes such as emerging diseases or climate stress, thereby increasing vulnerability to extinction in the long term. Number 2. Increase homozygosity. Among vertebrates, parthenogenesis commonly occurs through automatic mechanism in which meiosis is altered, leading the fusion of genetically similar chromosomes. This process results in increased homozygosity, where offspring inherit identical alleles for many genes. Elevated homozygosity increases the likelihood that deleterious recessive alleles will be expressed, which can reduce individual fitness, impair development, and decrease survival rate. The last one, genetic risk. The combination effect of reduced genetic diversity and increased homozygosity expose photogenetic population to several genetic risks, including inbreeding depression and the accumulation of harmful mutations because there is no genetic input from a second parent. Deleterious mutations cannot be effectively eliminated across generations. Although parthenogenesis may provide short-term reproductive advantages in isolated or low-density populations, this genetic risk limits its effectiveness as a long-term evolutionary strategy.
And next, we have advantage for species survival. First, sexual reproduction, which is Parthenogenesis. Parthenogenesis is a form of asexual reproduction where a female can produce offspring from fertilized egg without the presence of a male. Second, large body size and strength. Female Komodo dragons are strong and capable of defending themselves and their nest from predators. Third, long lifespan. A long life allows female to reproduce multiple times, increasing chance of species continuation. And next, we have ensure repopulation survival. Even a single female can start a new population, helping prevent extinction in harsh or limited environment. And lastly, efficient hunter and scavenger. They can hunt or scavenge efficiently, ensure enough energy for reproduction and survival.
In this slide, I'm going to talk about the example of parthenogenesis in other elements. There are three examples that I will give. The first one is honeybees, the second one is whip-tailed lizard, and finally, aphids. The first example is honeybees, or known as Apis mellifera. In honeybees, the parthenogenesis occurs through a process called arachnotoky. That is where the unfertilized egg will develop into haploid males, while the fertilized egg will become the diploid females, which is a worker or queens. This system will help to maintain the colony structure and efficiency. The second one is whip-tailed lizard. So actually, whip-tailed lizards are all female. So how does they mate, or how does they maintain their generation? So, this lizard will produce diploid egg that will develop into female offspring that have similar genetic to the mother. Even though male are absent, this lizard may show mating-like behavior that help to stimulate ovulation and increase reproductive success. Lastly, aphid. So how does aphid reproduce? So aphid reproduce mainly through parthenogenesis, especially during favorable environmental conditions. So the female aphid will produce live female offspring from unfertilized egg. Then it will result to an offspring that are genetically identical to the mother. So this will allow aphid population to increase rapidly in a short period of time.
Parthenogenesis has been challenged traditional reproduction concepts because parthenogenesis is an asexual reproduction carried out by vertebrate animal, where a female's egg develops into a new individual without being fertilized by a male's sperm. For decades, vertebrate reproduction was viewed as a process that requires sperm to create variation in the embryo. So, this led people to believe that hybrid animals would go extinct and not survive long in the world, because early biological theories and observations showed that most hybrids were indeed reproductively unstable. Some vertebrate species that can undergo parthenogenesis provide evidence of reproductive flexibility because females can switch to asexual reproduction, allowing them to establish new population without males and ensuring reproduction continues even when the sex ratio is skewed. Parthenogenesis is an important phenomenon in vertebrate reproduction because it serves as a means of conserving endangered species. This is so because parthenogenesis is an asexual reproduction, meaning it allows a population to continue producing offspring even without males. It also provides conservationists with extra time to restore habitat, increase population size, and manage the introduction of new males. Parthenogenesis also serves as a warning about genetic risk, as populations are more susceptible to diseases.